Hello everyone, welcome back. In this new chapter, we will talk about neoplasm. This chapter will deal with the basic concepts and general morphologic features of neoplasms and premalignant lesions. We will also discuss the principles for tumor nomenclature, differences between benign and malignant tumors, and comparisons between epithelial and mesenchymal tumors. This picture shows a segment of colon. We can see a large cauliflower-like mass projecting into the lumen. Microscopically, tumor cells form irregular glands with different shapes and sizes. Unlike the normal colonic mucosa, there is little evidence of mucin production. At high magnification, the neoplastic glands have clouded and darkly staining nuclei. Granular cells show larger than normal nuclei, some with prominent nuclei. Their nuclei are more varied in shape and size than normal, and the nuclear to cytoplasmic ratio is greater than that in non-malignant granular cells. The pathologic diagnosis is well-differentiated chronic adenocarcinoma. Here are some questions to think about. What is neoplasm and atypia? How do we distinguish between benign and malignant neoplasms? How do we name a tumor appropriately when we make the pathologic diagnosis? After this class, students should be able to define neoplasm and describe the gross morphology of tumor. Before further discussion, we should first understand some common terms. Neoplasm is a new growth due to abnormal, excessive, uncoordinated, and autonomous proliferation of cells. Even after removing the growth stimulus, the neoplastic cells continue to multiply. This means that neoplastic changes are generally irreversible once they have initiated. Therefore, neoplasm enjoy a certain degree of autonomy. In common medical usage, tumor is a synonym of neoplasm. According to their potential clinical behavior, neoplasms may be benign when they are slow-growing and localized without causing much harm to the host, or malignant when they grow rapidly, spread throughout the body, and may eventually kill the host. The common term used for all malignant neoplasms is cancer. The word cancer derives from the Latin word for crab, reflecting the true character of cancer since it sticks to the body part stubbornly like a crab. Oncology is the branch of science dealing with the study of neoplasms. Neoplasia is the disease process relating to the neoplastic growth. Neoplastic proliferation differs from non-neoplastic growth in several aspects. Firstly, neoplastic growth is virtually autonomous and independent of physiologic growth regulatory mechanisms due to changes in genetic composition. Reactive proliferation occurs in response to physiologic or pathologic stress, and this growth is controlled. Secondly, neoplasms are of monoclonal origin. Monoclonal means that the neoplastic cells are derived from a single mother cell. Thirdly, these neoplastic cells often form tissue mass that compress, invade, and replace normal tissues and organs. Therefore, neoplastic proliferation is uncoordinated and can cause significant effects on human health or even death. Finally, tumor cells exhibit different degrees of differentiation and often fail to achieve a highly differentiated state. To differentiate neoplastic lesions from the non-neoplastic ones, we should first study their general morphology. We usually describe the pathologic changes in terms of their macroscopic and microscopic appearances. Actually, the gross appearance of tumors may be quite variable. Certain distinctive features characterize almost all tumors compared to neighboring normal tissue of origin. We should give details regarding the number, size, shape, color, consistency of the visible mass. Tumors may be single or multiple in number. The left image shows a single mass projecting into the lumen of small intestine. However, in the case of the right, we can see multiple polypoid masses arising from the colonic mucosa. We can hardly tell the exact number. Please keep in mind that multiple tumors may represent the spread of individual tumor cells from the primary tumor. These are called metastases or secondary tumors. Tumor may range in size from small cell populations that can only be seen under the microscope to huge masses, measuring many centimeters in diameter and weighing many kilograms. As we can see in this microscopic image, the thyroid microcarcinoma is too small to be visible by our naked eye. We can only identify it under the microscope. Have you ever thought about what factors would influence the tumor size? In early stage, they are usually small, but they will enlarge as they develop into the advanced stage. Tumor location is another influencing factor. If the neoplasms arise on body surface or in abdominal cavity, the size can be very large due to the abundant space for the tumor to grow. For tumors developing in the cranial cavity, their sizes are usually small. If being large enough to be visible, tumors are often more or less round in shape, but any shape is possible. Some neoplasms cause surface defects, while others grow outward from the tissue surface. Special terms such as polypoid, papillary, nodular, lobulated, cystic, infiltrated, and ulcerated are used to describe the gross morphology of neoplasms. The shape of tumor is sometimes used to give diagnostic descriptions. The color of the tumor often resembles its normal counterpart. For example, tumors of the fat tissue usually look yellow, while tumors of blood vessels are red. Tumor tissue is often firm or hard because tumor cells tend to cause proliferation of fibrous tissue. And this is the reason why neoplasms usually cause palpable lump in accessible size. The composition and secondary changes determine the consistency of tumor. Neoplasms deriving from fat tissue are usually soft, while bone tumors are hard. Some tumors show hard inconsistency due to dystrophic calcification. If there are large areas of necrosis, tumors will be soft. In some situations, boundary between tumor mass and its adjacent normal tissue may help to predict its potential biologic behavior. For benign tumors, the masses are usually encapsulated and well circumscribed. Malignant tumors often grow invasively with indistinct margins between tumor and surrounding normal tissue. Now we have come to a brief summary for this section. Neoplasm is a new growth due to abnormal, excessive, uncoordinated, and autonomous proliferation of cells. In common medical usage, tumor is a synonym of neoplasm. Autonomous and uncoordinated growth, monoclonal origin, and failure to achieve a highly differentiated state are distinguishing features between neoplastic and non-neoplastic proliferation. In terms of gross morphology of tumors, we should give details regarding their number, size, shape, color, and consistency. Alright, that's all for this section. These are the references. Thank you.